and today I will talk about computational study of DNA silver clusters. So my talk will have three parts. First, I will give some brief introduction, what's our motivation to study this type of cluster, and also what kind of methods we use. And in second part, I will present most recent result we have is this DNA silver-4 cluster. And in the end, I will give some conclusion and outlook. So DNA is very interesting molecule because although it only have 4 bases, but it carry the genetic information of all the life. If we add some metal to the DNA, it can enhance the stability of DNA and add some new functional. So metal DNA actually is a promising building block for nanotechnology. In our group, we are most interested to study DNA silver cluster because these clusters have this sequence-dependent fluorescence. Which means if we choose a different DNA, it can form the cluster have different size, and they can have a different fluorescence color from blue, green, red to near air region. And also some of the cluster can have very high fluorescent quantum yield. On top of this, these clusters have a good photostability, low toxicity, low synthesis cost, and also DNA has a natural biocompatibility. So these clusters have many potential applications, especially for bio applications like biosensing, bioimaging, and also detecting the nucleic acid or the bacteria. But it's also very challenging to study this kind of cluster because it's very difficult to obtain the crystal structure from an experiment. Then if we don't know the structure and also the mechanism, it's very difficult to do the rational design of the clusters. Then if we not know the structure from experiment, how we study these clusters, the method we use is normally combine the QMM molecular dynamics with the time-dependent density function theory. First, we will build a system, have a DNA and cluster in the water. Then we describe the DNA atom and silver atoms by the DFT, and the water molecule will be described by first field. Because if we calculate everything by DFT, it will be too expensive. Then we run the molecular dynamics for several picoseconds. Then the silver will find the place they like. Then we will take some snapshots from this molecular dynamics. Then we calculate the spectra to compare with experiment. Normally, what we calculate and compare with experiment is this CD spectra. The CD spectra is the difference of the absorption of the left-handed and right-handed light. Why we calculate the CD? Because it's more sensitive than the optical absorption spectra, and it can show the small detail change of the structures. For example, before I come to Aalto, people from Olga's group, they have used this method to find the structure of this silver-mediate cytosine tetramer. They calculate the spectra and compare with experiment. They fit quite well, and they find in such system we have this interplanar hydrogen bond, which help to stabilize the silver atoms. Then when I joined her group, we also studied the silver atom with guanine tetramer, and we find one thing interesting is for cytosine, it have the right-handed orientation of DNA, but for this guanine, it have a left-handed orientation. So if I make a summary, for guanine silver tetramer, it's left-handed, and for the silver cytosine tetramer, it's right-handed. But both of them have the silver atoms bound to nitrogen atoms, and also DNA strands have a parallel conformation. So this is different than we have a natural DNA, because the natural DNA have this anti-parallel conformation. After this study, we begin to know more about the interactions between silver and DNA. So we want to discover what kind of cluster silver will form with DNA. There are not much direct information from experiment, but from experiment we know some key facts. The first fact is from experiments, they know these clusters have both silver cation and neutral silver. Then the second key fact we know from experiment is they know the cluster most likely to have this long robe shape. And the third fact they know is they know it exists some kind of a magic number, like these ligand prot clusters, they also have a magic number. And for DNA silver cluster, the magic number is 4 and 6. So these clusters like to have a neutral silver atom, 4 or 6. And another thing we know from experiment is the fluorescent clusters like to have the cytosine-rich or the guanine-rich DNA templates. Based on all this experimental fact, then we build our model. We choose a DNA sequence have 6 pair of cytosine, because the fluorescent ones like to have a cytosine-rich sequence. Then we choose the magic number 4, silver neutral, and we use different number of silver cation from 2 to 4, and we keep this parallel DNA configuration from the previous calculations we found. Then we first run 2 nanosecond classical MD for equilibrium the system. Then we run 6.5 picosecond QMMD. Then we connect the structures from the last 2 picosecond for analysis. Okay, so here is the structure we find from simulation. We can see from left to right, they can form silver 4, silver 5, and silver 6 cluster in DNA. And they all have this 2-row and a planar structure. If we zoom in, we can see many of the silver atoms are bound to nitrogen, and there is no silver oxygen bond, which means in the DNA silver clusters, the nitrogen play a central role to stabilize the clusters. Then we calculate the CD of these clusters. Then we find they all have first positive peak around 2 eV, and the second peak negative around 3 eV. But for this silver 4, the first peak is very small. I want to mention that because for this DNA silver clusters, they are very dynamic. So it's not enough to calculate one CD. Instead, we should capture some snapshot, and we calculate the average CD to compare with experiment. So here we collect 5 structure and calculate the 5 CD and average them. To make sure the 5 structure is enough, we change the site we choose, and we can see they all present the similar feature. Then we are confident these 5 structures enough to show the dynamic property of the clusters. Okay, then after we get the CD, we compare with the experiment. We can see although the experiment have different DNA sequence, but it's very often they also get this kind of CD spectra, have the first positive peak around 1.5 to 2.5 EV, and the second peak around 3 EV negative, which fit very well with our CD. Then we think this kind of planar 2-row structure should be commonly exist in the DNA structure. When we were doing the simulation, there were no experiment results yet, but when we were doing this work, the first ever crystal structure of silver DNA was reported, and in their structure they also find this 5 silver cluster, which is very similar as this 5 silver cluster we found in our simulation. So we are very confident our prediction is correct. Then we also study the charge of the cluster. We can see the silver atoms in the different position of DNA have different charge states. On the top and on the bottom, they are more like silver cation because they have the charge close to 0.6, close to 1. Their function is more like a glue. They will stick these 2 DNA together, and in the middle, they are more like neutral silver. And if we add them together, they will about have 4 electrons. So they are all this magic number silver 4 clusters. Okay, then besides the CD, we also calculate the optical spectra, and we found in the first peak position of CD, they also have a peak from optical absorption spectra. Then we think if we analyze this peak, it will help us to understand what this chiral optical property come from this cluster. So we calculated the density state of the cluster. Here is the occupied state, and here is the unoccupied state. Then we calculate the transition map of this peak. Then we find that this peak is due to the electron transfer from the state here, which is mainly have the silver, and to the state have a lot of contribution from DNA. So this optical property is because the transition from silver to DNA. Then if I make a conclusion, we find silver atoms in DNA silver 4 clusters have a 2-line planar shape, and there are silver nitrogen bond, but no silver oxygen bond in the cluster, which means the nitrogen atoms play the central role in the stabilization. And also we find the original of this CD signal. It's from the CD DNA transitions. And for the outlook, as I mentioned before, besides this silver 4, they also have another magic number, is this silver 6. So we could also study the silver 6 cluster. And also beyond cytosine, we also have other DNA templates like adenine, guanine, and thymine. So we can also study what will happen if we have different DNA template. And the last thing is because this most interesting property of these clusters is it have the fluorescent. So our optimal goal is like, can we understand better why these clusters have the fluorescent and how we can choose the optimal DNA template to make them have a most strong fluorescent signal. So thanks for your attention, and I'm willing to answer the questions, and I also hope we can discuss anything related to the cluster or the simulations. Thank you.
Thanks, Professor, for your presentation. Now it's time for the questions, comments. Please go ahead, Manuel, with your question.
Hi, Professor. I have a question about the software that you employ in the development of your calculations. What kind of software or what is the name of the software that you employ?
Yes, so for the molecular dynamics, we use this CP2K, and for calculating the circular diagram, we use GPA normally.
Thank you very much.
Another question, comment?
I have another question. When you talk about the clusters of 6 silver atoms, what is the what is the implication in the real biologic system? What is the possible application for that kind of cluster?
Many applications are actually related to this fluorescent, because if you want to have some bioimaging application, you need to have a kind of like increase the visibility of, for example, the tumor or some virus or something. So one thing is in this bioimaging, and also like the biosensor, because for example, you have some different DNA, and then some of them can have the fluorescence, some are not. Then you can detect if here is some DNA mutation or not. So mostly for this bio applications.
Thank you very much.
I have a question.
Yeah.
Why you are using just 3 nucleobases and not 5 nucleobases? What is the reason you choose between these 5 nucleobases?
I didn't understand where the 5 and
because you are using your cytosine one in, I'm not sure, CG and maybe T, right? But you have 5 nucleobases forming the DNA strand.
Okay. Because if we mix different type of DNA bases, it will make the calculation very difficult. Then it have like many possible places where the silver will bound to. So for now we have only only 2 bases, we think. One is cytosine and one is guanine, because this is mostly experiment to find when you have a lot of cytosine or guanine, they have this strong fluorescent. So we didn't include other bases in our simulation.
But that makes sense just using 3 of the 5 nucleobases. So, I mean, there are some structures just containing these 3 specific nucleobases or not.
Yes, yes. I mean, in the lab, you can also make a DNA strands only have one basis. It's also okay. Like you can control which kind of DNA basis you have.
You mentioned that when you was finishing your work, the crystal structure was reported, right?
Yeah.
So maybe you get the positions from that crystal and repeat the calculation and compare against your prediction, for example, or not?
Actually, not, because when we saw this paper, we already finished our manuscript, so we didn't have had a look when we do our simulation. But what we try to compare when we do the simulation, actually we try to compare the another experiment work. They reported this CD spectra. That is what we try to compare when we do this work.
That negative and strong peak located near to the 3.5 eV, do you think that is displayed by a small cluster, or what's the reason of that peak, negative strong peak?
Yeah, that's a very good question. Actually, we didn't analyze that peak. Yeah, it's a very good point. Maybe we should also analyze this peak, because we only analyze this first peak. It's from this silver to DNA transition, but this one we didn't check yet.
Because we have, for example, that the strong peak and negative sign in the gold 15. And these are small clusters, so maybe there is a similar, I don't know, explanation or something like that.
Yeah, that's very good suggestion. Maybe I should have a look, like what is the contribution for this peak. Yeah.
And you mentioned that the first peak in the UV or CD spectra is due to the HOMO-LUMO gap, right? The transition among the DNA and the silver cluster.
Yeah, views.
So we have
yes.
We have a mixture of these contributions coming from the silver and from the DNA molecule, right?
Yeah.
And that's happened when you have small clusters, small silver clusters, for example, the contribution is major or is large with respect to the big ones.
Yeah, I think for the first peak, it's very common from the HOMO to LOMO, but for the second peak and the third peak, it could have a multiple contribution, like not one orbital to another, but it's like could be several orbitals contribute to that one.
Okay, thank you. More questions, comments? Maybe you can, all right. Diego, go ahead with your question.
Oh, yes, I have a question. Thank you, Professor, for your presentation. And I wanted to ask a question about the conditions of the simulation. The temperature, for example, under which conditions was it run?
Yeah, I think it's better I go back to that page. Yeah. Yes. So for the molecular dynamic, we use this room temperature, like 300 Kelvin, with this NVT condition in the molecular dynamic. And when we calculate a spectra, we just have zero temperature and in the vacuum.
Okay. Since these biological molecules are very sensitive to these conditions, right?
Yeah. Yeah, that's, I think also it's different than the large cluster. If we have one large silver or gold hybrid cluster, we can just optimize the structure and we calculate the spectra to fit with the experiment. But for this type of small and dynamic cluster, we need to run the long simulation, and we kind of like take 10 or 20 or depends your system, and then you calculate the average spectrum.
Okay, thank you. And well, I wanted to ask also about the number of particles that are included in the simulation.
Yeah.
More or less, how many?
Yeah, for silver is I think from 6 to 10. Depend different cluster we use, and for DNA I think it's about 200, so it's around 200 atoms in the whole simulation.
Oh, wow. 200, and the simulation is run in parallel. I mean, how do you say, processors?
Yeah, yes. The most expensive part is this molecular dynamic simulation. For the CD simulation, it's not that long, but need like some parallel calculation. Yeah.
Okay. And I see the picture, and I understand there are like explicit solvent conditions, right? That is the
Yeah.
molecules of the solvent. Oh,
Yes.
and those are some expensive calculations. Okay,
Yeah,
thank you, Prof.
because as you mentioned at the beginning, for this kind of systems, it's important to include also a bit environment because they are exist in the water, right? So, yeah.
Oh, okay. Then using implicit solvent conditions wouldn't be, I mean, as optimal as simulating them explicitly.
Actually, I didn't check that. For example, if we have this implicit water, how it will happen. I think I have this collaboration. Olga Lopez, actually, I think she said she will test some like if we have some this background water like implicit, how the dynamic will go. We haven't checked that yet.
Ah, okay. Yes, because when including those specific atoms or molecules, it becomes more expensive. We are working on something with how is the word, short chains of polymers, and yeah, using implicit solvent makes it more faster the simulation, but I think some information is lost, right?
Yeah. I mean, it's depend how important this water in the system, right? If it's very important role, then it's good to use some explicit model. But if it's just more like a background, then I think the implicit water is enough.
Okay, I see. Very interesting. Thank you, Professor.
Yeah, thank you.
Okay, more questions? I got one.
Yeah.
I got one. And this is related to Diego's question. You make these molecular dynamics calculations to simulate the experiment conditions, temperature, solvent, but after that you get these positions for the silver atoms and the more close nucleobases, right?
Yeah.
And you make in vacuum the calculation for the CD spectrum.
Yes.
How many atoms you are using for these calculations of the CD spectrum?
It's depend on the code. Like for this GPAL code, we can have like a 200. It's still okay. Even we have some system with 1,000, it's okay. But when we use Gaussian, then it's maybe need to be within 100.
So how long those calculations take to make done?
If we have 200 CPU, maybe it's take 2 days, 1 or 2 days.
Okay, that means that the GPAW is fast.
Yeah. Another thing is we are not using very expensive function. We just use GGA type of function. The reason we use Gaussian is we in one point we want to check how the hybrid function behave. So use PBE is much cheaper, and it seems okay for this system.
Okay, and GPA use something like plane waves.
Yes, it's like a VASP. It's use this PAW approximation for the plane wave. So.
Okay, thanks, Manuel. Go ahead with your question.
Thank you. Related to the DFT calculation, can you explain me what kind of basis set do you employ in this calculation? I mean, if you run with explicit core function or pseudo potentials, what kind of basis set do you use in this?
For this molecular dynamic, we use this some default basis sets in CP2K, but for the moment I don't remember the name of the base set. And for water, we use this amber force field.
Thank you. In the case of Gaussian, what kind of calculation use separate potentials? I mean, from the silver atoms to the nuclear bases. Is possible to do different pseudo potentials in Gaussian?
We didn't apply this technique. We just use the same base set and the same function for Gaussian.
Okay, yeah. Thank you.
Yes, thank you.
You mentioned that it's necessary to make an average of the conformations for this structure, right? Because it's not just possible to use one of those conformation or configuration, right?
Yeah.
You use around 20 conformations or more than that?
For this small system, like for tetramer, this receiver, in this picture we use 10, and for the larger ones we use 5. But actually we normally calculate 15, because then we can check all these 5 give same result. Or if we average 10, we choose 30, then we can see all these 10 average give same result.
Okay, thank you. More questions?
Yeah, that we choose different site to compare.
So how long you take to make these calculations? I mean, all the time that you spend in this project.
Oh, that's that's depend. I think when we find the structure, one structure checking is not very long, maybe like 1 month. But it's very difficult to find the correct one. Because they have so many possibilities where the silver could be.
Okay. Thanks again for your answer, and maybe we have more questions, comments from the audience.
I have a comment, Professor.
Yeah.
I think that these kind of calculations in general could help to understand the DNA damage caused by heavy metals or metals in general. It's possible to understand that kind of damage of DNA using this type of calculations.
Yeah, I think so. I mean, our specialty is not on that field, but I think if someone want to study the damage of this toxical ion to the DNA, it could use the same technical, like the QMM simulation, and yeah, sure.
Okay.
No more questions? All right. Thanks again for your nice talk, Professor Chen.
Yeah, thanks a lot for inviting me.
It was a pleasure to hear about your work, and maybe we can contact, have some talk after this webinar. So thanks again.
Yeah, okay. Yeah. See you.
All right.
Yeah.
Have a nice day.
Yeah, have a nice day, everyone. Bye.
Bye-bye.
Bye.